Fractal dimension is a non-scalar measurement of the complexity of a fractal. A small fractal dimension denotes a lack of complexity, while a large fractal dimension indicates otherwise. Lacunarity is a geometric measurement that describes the texture or size of gaps in a figure. Lacunarity can also be thought of as an indicator of rotational variance. The more affected the figure is by rotation, the greater the lacunarity. Figure A looks relatively unchanged by the rotation, while figure C looks like two completely different figures before and after the rotation. Thus, figure C has a greater lacunarity. There are two different types of sets that can be used to explain the complexity of a fractal: Julia set and the Malbrot set.
Tumors have a higher fractal dimension than normal tissue, indicating their greater internal complexity. Simple Euclidean geometry does not prove useful when assigned the task of describing something as chaotic as a tumor. Fractal geometry enables people to further analyze the growth and development of cancer through the fractal dimension of fractals.
First, we look into the brain to discover its fractals. In this slide, we see two pictures of the human brain. To the left, there is a picture of a healthy brain that has been sliced horizontally. The brain has visible fractals, as shown in the picture, but it also has underlying fractals in the blood vessels of its tissue. To the right, there is a picture of a brain with a tumor in the center. The white blotches in the brain are the tumor. The tumor does not have visual fractals, but instead the fractals can be found within its tissue. Also, the tumor grows in a fractal manner. An anagram, CT scan, magnetic resonance imaging scan, and diffusion tensor imaging are all used to locate tumors in the brain. The fractal model has been proven to be useful in analyzing a wide variety of medical images, including brain scans. Brain magnetic resonance images are candidates for characterization using fractal analysis because of their high complex structure. The box counting method is used to measure the fractal contour of the tumor cell. The box counting method is based on counting the number of boxes having side length r needed to cover the surface of a fractal object, and the number of grid boxes n occupied by one or more pixels on the images, shown in the diagram to the right. The first column shows images of healthy brains without any tumors. The second column shows the same brains now with growing tumors. The last column provides a depiction of the box counting method oncologists use to measure the fractal dimension and fractal growth of the tumor. The white boxes are where the tumor initially starts, and the lighter gray boxes show the gradual growth of the tumor. Over time, as the tumor gets bigger, the number of boxes will increase and the rate of the tumor growth can be determined.
The first picture, image A, shows a close-up of healthy brain matter. The fractal dimension of healthy tissue in a brain is 2.3. The second picture, image B, shows a close-up of the same brain matter now with a malignant tumor growing. The fractal dimension of the brain matter is 2.5. Image B was taken 3 months after image A was taken. The last picture, image C, shows the same tumor 3 months after image B was taken. The fractal dimension of the tissue is now 2.8. As shown in the graph to the right, the fractal dimension of the brain matter gets bigger as the tumor grows. The fractal dimension differs between normal and tumor images, ranging from 0 to 0.06 in normal tissue and 0.04 to 0.457 in tumor images.
Finally, we'll be discussing hepatocellular carcinoma, which is also known as liver tumors. The liver is a large lobe glandular organ in the abdomen of vertebrates, involved in many metabolic processes. These are two CT scans between a cancerous liver and a healthy liver. The normal liver is free of tumors and is one whole mass, whereas the cancerous-filled liver is being invaded with two dark masses, where the fractal dimension starts to form and the cells start to rapidly multiply, making it more and more harder to cure as the fractal dimension multiplies and the tumors grow within the body. Table 1 shows basic statistical parameters on fractal dimensions of normal malignant nuclei. The change in fractal dimension showed to be 70, and the change in the mean was 0.04778. They compare the fractal dimension of malignant nuclei in the liver, and there's a clear change in the fractal dimension because the development of tumors take up a higher space in the body and invade all the healthy cells. In Table 3, they calculated fractal dimensions of some geometrical figures versus fractal dimensions determined by present system, which is the liver. So it's comparing daily items or figures to the liver just to show how extreme the tumors are. The frequency distribution of fractal dimensions of malignant cells are centered more towards higher fractal dimensions, whereas the frequency distribution of fractal dimensions of normal hepatocytes are more evenly distributed and the mean is not centered as high. Notice how the cells in the malignant HCC cells break off and become separated into smaller fragments. On the left is isolated nuclear shapes of normal liver cells, and on the right are isolated nuclear shapes of HCC liver cells. This can be seen as an iteration because the cells start to multiply as the tumor starts to form. Part A shows a biopsy needle inserted into the liver and tumor. As you can see, as the tumor starts to progress in this area here, the fractal dimension starts to multiply, as it does in Part D as well. Part B and C also show a close-up of the liver.
The lung functions as a source of oxygen for human beings. When the blood vessels that control a human body's flow of oxygen stop functioning, it is most likely due to the cancerous cells that have invaded the respiratory system. Fractals in the lung help increase the efficiency of the organ. They maximize the surface area of the lung so gas exchange can take place more productively. The actual surface area of the lung is approximately the size of a tennis court. In human lungs, there are 11 orders of branching, from the trachea to the alveoli at the tips of the branches. Here's an example of the fractal structure of a human lung. In Figure A from the photo above, blood from the heart moves into the lungs in the blue blood vessels. The blood becomes oxygenated in the red blood vessels and eventually is pumped away from the lungs. The blue vessels branch into smaller parts because smaller branches allow for a larger surface area. Thus, more blood can be oxygenated in the red blood vessels. The branches come together towards the top of the red section, allowing oxygenated blood to be pumped to the rest of the body. The other figures show what blood vessels in a cancerous lung would look like. This data shows that cancerous lung tissue has a higher fractal dimension and a lower lacunarity. Categorizing different patients based on their fractal dimension and lacunarity can prove useful in the clinical diagnosis of patients with lung cancer. The larger the fractal dimension, the more dangerous the tumor is. The image to the left shows a patient's pre-treatment tumor CT, and the image to the right shows the same patient's post-treatment tumor CT scan. After treatment, the tumor decreased in mass greatly. The analysis of fractal dimension of lung tumors has allowed scientists to target specific aspects/areas of lung tumors and successfully reduce the size of tumors. The CT scan above clearly shows the size of the tumor has decreased significantly.